Hello, everyone. I'm here today to talk about a project that we've been working in our lab, which is focused on multimodal machine learning for dementia assessment. This is an effort by a multidisciplinary team of people in our lab. As you can see on the screen, we have a variety of students and postdocs with various backgrounds, some of them getting their PhDs in computer science, and some of them who are postdocs who are working at the multidisciplinary level. We have some MD students who also collaborate with our PhD students and postdocs, and together we are very excited to work on this important question, which is related to applying machine learning and developing novel machine learning methods to process routinely collected clinical data to understand the neurodegenerative condition, which is dementia. So I want to start off by briefly talking about the questions that we address in our lab. Like I said, we focus on methods development, and our methods development is mainly driven by trying to understand how we can develop machine learning or deep learning methods that are interpretable. This is a very attractive question to ask because a lot of fields, not just in medicine, a lot of fields are interested to understand how a machine learning model works and how it can be interpreted. Also, lucky for our lab, we're able to attract basically those mainstream computer science students because they're very motivated to come up with some innovative solutions to address the interpretability aspect. We are also equally motivated to apply this to the field of Alzheimer's disease and dementia, because in the kind of goals that we set are mainly to build models that can use routinely collected clinical data, and those that can assist a clinical or a clinician setting or an end user. We are mainly interested in medical imaging, although we spend a lot of time to process other forms of data, uh, such as demographic data, cognitive test results, and many other known correlates of disease. So if we have all these multiple modalities of data, then we want to think about developing multimodal machine learning algorithms. Most importantly, because we interact and learn from the clinicians, we don't want to just build fancy models, but we really want to spend a lot of time to figure out ways to validate our models and model-based findings. Um, Clearly, for us to do any of this stuff, we need data. And we've been very fortunate to collaborate with some senior investigators at Boston University and outside who generously guided us to provide access to a lot of data sets, as you can see on the slide. And most of these data sets are actually publicly available, so anybody who's interested can download this data. You just have to complete a form and then submit the form, and then you'll be able to download the data. But only at the one at the center here is the BU managed data set, which is called as the Framingham Heart Study, which is about 15,000 individuals living in the town of Framingham in the United States. And we've been collecting data on those people. As you can see from this slide, there are lots of different participants, and if I count all the numbers, it becomes a very big data set, tens of thousands of participants. But what's more important is that all these data modalities are available. Like I said, the demographics, the patient level history, the imaging, etc., are available from all these cohorts. And if you download all this data, and if you're thinking about asking interesting questions, then we are thinking about multimodal learning. Obviously, the data set becomes huge as well. So as you can see, counting all these participants and all the multimodal data, we're talking about terabytes of data. So you need, obviously, the needed computing infrastructure to work on these things. I also want to take a minute to talk about another unique cohort that we have. So Boston Medical Center is affiliated with Boston University. Here we have data coming from diverse populations, which means we are able to sort of look at variabilities within genders, diversities, etc. So given all this data again, from all these multiple cohorts. And because we were very fortunate to have computer scientists work with us, one of the first things that we started doing was to develop computational frameworks that allowed us to automatically process this data. When it comes to images, things become a little bit more tedious. You need to do a lot of things to pre-process the data, such as register these images to a predefined template, perform simple quality checks just to make sure the scan has data or contains artifacts. If you find artifacts or simply unwanted aspects, then we have to figure out a way to remove them, or maybe select certain regions of interest within the scans. And then we basically focus on specific analysis. If we want to process data from multiple cohorts, then we need to also figure out a way to normalize the data or harmonize the data. And these aspects are not really trivial because we are dealing with different imaging modalities such as MRIs, PET scans, and within MRIs, we are also thinking about different scanner level effects. So we need to really think about aspects such as bias correction, etc. To be honest, these things may not sound that important if you're dealing with, let's say, 50 or 100 or 200 cases. But if you have 50,000 cases, then you need to really start paying attention to these aspects more carefully. In our lab, we spend significant amount of time, in fact, more than 50% of the time, as you can see on the slide, with just data processing. And once you process all this data and harmonize everything, then we really think about deep learning or building some machine learning methods on the data that is being collected. Let me show you some example. So here, as you can see, this is we created a database of MRIs, and then there are several ways to sort of process the data. And the first thing we do is we do this registration, a volumetric registration. And there are many image registration methods that are available. And we used a framework called as an MNI 152 template, which is basically a template provided by McGill University in Canada, which allows us to register all these scans. And there is another tool provided by Oxford University, which is called as FLIRT tool, FLIRT, FLIRT tool within a package called FSL, which allows us to sort of really align the scans with respect to that MNI template provided by McGill. One thing we realized is that none of these automated registration techniques, they don't work really well for all cases. So which means for those cases that are not registered, we then go back and then do some manual operations to make sure that we do some simple affine transformations or perform manual registration, and then use certain things as landmarks to register these images. And then once we register those images, then we kind of normalize all the intensity values, then we remove outliers, we adjust their intensities, and then we perform background removal. And when I say background removal, I'm also talking about something called as this stripping of the skull, which is important because the skull is very bright on MRIs, and we don't need that information essentially to sort of look at what's inside the skull. After all this, we end up with the actual imaging data that is actually amenable for machine learning or deep learning. And we usually save this data in numeric formats and process them on our computing servers. One of our main motivations was to see if we could develop and validate an interpretable deep learning framework that could use readily available data such as imaging, demographics, bedside cognitive testing to assess if a person has Alzheimer's disease. It all started when a PhD student named Shangran, the first author of this paper, came up with an idea of creating a computationally efficient deep learning framework to process raw MRI scans of the brain. This deep learning model is a fully convolutional network, which is a variant of the traditional convolutional network that I described, that you probably know. Shangran and his team kind of developed this efficient neural network that takes these volumetric patches of the raw MRI scans and outputs these volumetric heat maps that kind of nicely highlight the regions or the subregions within the brain that point to a high degree association of disease risk, which was then used for making that final prediction. With this kind of framework, one can visualize these high-risk subregions. And this was kind of an innovation that was highly appreciated by the reviewers. From the computational standpoint, this framework can work very efficiently because it can process these volumetric scans very quickly, as the model was trained on those inferring local patterns of the cerebral structure that kind of suggested the overall disease state. Then we developed the model using the data that we got from a cohort called as Alzheimer's Disease Neuroimaging Initiative. And then we took a lot of time and processed data coming from three other cohorts, which is, as you can see here, one is called as ABLE, which is a cohort from Australia. And the second column here is the data from Framingham Heart Study, which is from Boston. And the third column is the NAG data set, which is coming from multiple different Alzheimer's disease centers in the US. So the model was trained on the ADNI cohort, and then it was tested independently on these three different cohorts. And the reason we did this because we wanted to make sure the models that we build on one cohort are generalizable on the other cohorts as well. And this kind of really serves as a computational validation of training a model on one cohort and then making sure external data set is used for testing the cohorts. And then in a follow-up study, we asked a slightly more interesting question where we are again collecting data from multiple different cohorts. But in addition to just looking at MRI imaging and demographics and single cognitive test here, we extracted much more information from these participant records, such as demographics, an extensive neuropsych battery, functional assessments, past medical history, medications, and as well as MRI scans. And then we came up with this approach, which combined information from all these different modalities of data, and then tried to ask basically two questions. The first question was, given all this data, can you assess if the person has healthy cognition, mild cognitive impairment, or dementia? And if the model predicts the person to have dementia, then is the dementia due to Alzheimer's disease or due to some other conditions or some other causes? And then again, Shangran and his team here came up with the interesting framework, which allowed us for this multimodal data fusion. And after building the model, we also took a while to make sure that the model was validated. So we did three different things. And the first thing that you got seeing on the right side is we took the predictions from the model, and then we saw their associations with neuropathology data, which in this field is considered as gold standard evidence. And then we recruited a bunch of clinicians. Some of them are neurologists and some of them were radiologists, to make sure we performed a head-to-head comparison of the model findings with respect to the neurologist or the neuroradiologist assessments. I'm going to go over this in a little bit here. So again, quickly summarizing. So when I say multimodal data, I'm talking about demographics, patient history, functional assessments, neuropsychological testing, MRI scans. And in total, we had about 4,500 participants with healthy cognition, about 2,400 participants with mild cognitive impairment, which is a stage before the person is expected to see that they have dementia. And then 1,600 participants with dementia due to Alzheimer's disease, and about 348 participants due to having dementia due to some other causes. And on the right column, you can see here is that we collected data from multiple different cohorts. I think this was the most important step and the most time-consuming step, because we wanted to make sure the models built on one are generalizable across the others. So this, I think, added a huge strength to our study. And then once the data has been collected, we went through these two steps. And the first step being, leveraging a convolutional neural network, which allowed us to process the MRI scans. And then once the information came from the MRI scans, we fused that information with the non-imaging data using a multi-layer perceptron, which allowed us to combine sort of this vectorized information from the imaging data and the non-imaging features to sort of make those assessments. After we performed the model, one of the first things we did was we sort of understood, or at least tried to quantify, what factors, what imaging or non-imaging factors contributed to the actual label of interest. And on the left side, you can see is the task, which is related to healthy cognition, mild cognitive impairment, and dementia. And then this is basically a rank ordered list of features that turned out to be important in terms of making that classification. And if you talk to an expert in the field, they would somehow agree to the fact that the top feature that turned out to be important is called as MMSC, which means mini mental state examination, which is a very common and standard bedside cognitive test that is performed to assess a person's cognitive status. So that was reassuring for us that the importance analysis using SHAP allowed us to identify the most important factors contributing to that output of interest. And on the right side is again the rank order list of features that turned out to be important in the context of assessing the next question, which was, is the dementia due to Alzheimer's disease or due to some other reasons? And interestingly, the top factor that contributed to this prediction was MRI, which was imaging data coming in from those multiple different scans that we collected, right? So these things sort of allowed us to, or at least gave us confidence that our model was doing something reasonable. And then Changran and his team also tried to go back into those MRI images and then try to identify regions within the MRIs that turned out to be highly associated or had higher risk of making that prediction. So in this context, certain regions, as you can see, were highlighted. And these regions were classically known to associate with disease such as hippocampus, amygdala, entorhinal cortex, etc. So these kinds of interpretability techniques, I think, allow us to, again, get confidence that our model is doing something reasonable and it sort of falls within the expected clinical observations that people have already known. So this is kind of, again, reassuring for us. And then for about 100 cases or so, we had data coming in from people who died and their brains were autopsied and then analyzed. And from those regions where the analysis was done, the grades of disease were estimated by expert pathologists. So what we did was we took that information and then we tried to associate our model predictions or model predicted probabilities with the grade of disease. So here what you're seeing are these three different plots indicating what classically I think pathologists do to assess Alzheimer's, which is looking at amyloid beta score or amyloid beta grade disease. And then the second is Braak and Bach staging, which is about looking at a protein called as neurofibrillary tangles. And the third one is the CRAT score, which is again a very commonly used way to assess the disease in the brain. And using standard statistics such as one-way ANOVA test, we rejected a null hypothesis that of there being no significant differences in these model predicted probability scores between semi-quantitative neuropathology scores groups with a confidence interval of about 0.95. Again, this is again very reassuring for us to see that the model was trying to predict the disease accurately using all that information. And then finally, we identified like a few cases from the Framingham Heart Study where the neuropathology reports were available. And these reports basically included, region-specific scores of different proteinopathies, which is basically the protein depositions indicative of Alzheimer's. So what we did was we sort of used those interpretable heat maps from our model and then overlaid them on the regions where neuropathology scores were available. What you're seeing here on the slide is just basically the overlay predicted by the model, overlay of the model predicted regions of high Alzheimer's disease risk with postmortem findings of Alzheimer's disease pathology on a single person. This person actually had clinically confirmed Alzheimer's disease with affected regions, these temporal lobes and the hippocampus and the cingulate cortex and the corpus callosum. The first column basically shows the MRI, and the second column shows the heat maps generated by the model, and the this third column shows basically like an overlay of those things, and the fourth column shows the pathology. And the fifth column is basically overlay of the pathology and the model predictions. Again, this served as a very important way to validate our model as well. And then finally, the icing on the cake, like I said earlier, we recruited about 17 neurologists, and they were provided with 100 cases that included all that information that the model saw, which is imaging data, demographics, medical history, neuropsych tests, and functional assessments. And they were asked to provide basically the diagnostic impressions of who in those 100 cases had normal cognition, MCI, or Alzheimer's disease dementia, or dementia due to some other regions. For within those 100 cases, we had about 50 confirmed cases of dementia. And we took those 50 cases of dementia and then we gave them to the neuroradiologist, but we gave them limited information. We gave them just the imaging data, age, and gender. And then we asked them to provide diagnostic impressions of whether the person has dementia due to Alzheimer's or due to some other reasons. And interestingly, our model seemed to have worked pretty reasonably well. And as you can see here, the first row shows the performance of the model, which is the green line. And the dots basically represent the performance of the neurologists, 17 of them. And our model seems to be doing slightly better than the average neurologist. And the second row here shows the performance of the radiologists. And the red line is the performance of the model, and the dots are the performance of the radiologist. And again, the model seemed to be doing reasonably well compared to the average radiologist. So this is kind of really what I really wanted to talk. Essentially, the goal of our lab is to build interesting machine learning methods using routinely collected clinical data, which can be multimodal data. And we have some really talented students and postdocs who have a variety of skills who are interested to sort of really create some impactful solutions that can assist clinical practice and beyond clinical practice. And, maybe, tomorrow we are thinking about building these models to assist clinical trials, right? So there are lots of different opportunities for AI in medicine. And we've been very fortunate to sort of work in such an important question, which is related to dementia. And I think we are also very fortunate to have data coming in from multiple different cohorts across different collaborations. And I think there is an exciting possibility here for us to sort of really bring AI-driven tools to the clinic. But before we do that, I think we need to sort of really think about a few aspects, and that I'm summarizing here. The first thing is, when we start thinking about data, we need to really think about how to process the data in the native formats. We need to collect data, first of all, in native formats, and we need to also think about how to harness that raw data, because I think it has the most information. And given the recent and breathtaking advances in machine learning, I think we have an opportunity for us to really tackle interesting questions in medicine. And with respect to the machine learning standpoint, I think there is an opportunity for us to think about processing multimodal data, which is basically the data that normally a clinician would see in a clinical setting, and use that data to see if we can come up with some interesting solutions. And to be successful, we are still trying to be, we're still trying to get there. But I think things to keep in mind is that, it's not just one group of people who can get this thing done. It's about partnerships, it's about collaborations. And we've been very fortunate to actually collaborate with the multiple different experts in different fields, including computer scientists, clinicians, neurologists, radiologists, neuropathologists, neuropsychologists. All these people, I think, can help and then can provide important insight into the question. And I think together we can sort of hopefully come up with interesting solutions. And what we also focus importantly is on not just on method development. We also want to make sure that once you build these fancy machine learning models, they have to be validated. Only when you validate them, the end user per se, which is, let's say, the clinician, is going to be able to appreciate it, because they want to make sure there is strong evidence behind all these model predictions. So finally, I think, like I said earlier, the promise of AI can reach the patient, or the tools that we build can reach the patient only when we actually have these collaborations, and only when these clinicians or these healthcare providers adapt these tools, which means we need to make sure these tools are validated and understood by these clinicians so that they can actually adapt them. With that, I really want to thank my sponsors, including funding agencies coming from National Institutes of Health and various other organizations. And thank you for your time.